Every organism must transmit its genetic instructions to the next generation to survive. Understanding these precise cellular mechanisms is a mandatory requirement for the MCAT. In fact, questions targeting reproductive biology and cell division account for roughly 9% of the entire biology section on the exam. To master this material, we have to track the process across 3 distinct scales. We start at the microscopic level of DNA replication, scale up to the specialized organ hardware that builds the gametes, and finish with the chemical software that controls the entire system. Memorizing vocabulary words is not enough here. Scoring in the top percentiles requires a strict mechanistic understanding of how these biological layers interact and exactly what happens when they fail. Most of a cell's life is spent in interphase. During this period, DNA exists as uncondensed chromatin. The genetic material must remain in this loose, unspooled structure so that RNA polymerase can physically access the genes for transcription. This diagram illustrates the standard cell cycle. Actively dividing cells flow through 4 stages: G1, S, G2, and M. Non-dividing cells exit into an offshoot pathway called G0, carrying out baseline functions without preparing for division. The transition from G1 to the S phase is governed by a strict restriction point. The cell must verify that its DNA is undamaged before it commits the energy to replicate its entire genome. The primary enforcer at this checkpoint is the tumor suppressor protein p53. If it detects structural damage in the DNA, p53 arrests the cell cycle entirely until repairs can be made. Once cleared for synthesis, the cell duplicates its genetic material. A common point of confusion here is the chromosome count. Copying the DNA does not change the overall ploidy of the cell. A human cell entering the G2 stage still possesses exactly 46 chromosomes. The difference is that each chromosome now consists of 2 identical sister chromatids, joined at the centromere, bringing the total chromatid count to 92. The cell cycle operates as a heavily gated pathway. If a physical checkpoint fails, often due to a mutation in the TP53 gene, damaged cells divide rapidly and without restriction, resulting in the unchecked tissue accumulation we identify as cancer. For routine tissue maintenance, the body relies on mitosis. The strict objective of this process is to produce 2 diploid somatic cells that are genetically identical to the parent, preventing any loss or alteration of the genetic blueprint. The division begins with prophase. The loose chromatin condenses into tightly coiled chromosomes, while centrioles migrate to opposite poles to form the microtubule spindle apparatus. During metaphase, kinetochore fibers interact with the spindle apparatus to push and pull the chromosomes until they align perfectly along the equatorial plate in the middle of the cell. Anaphase provides the actual separation. The centromeres snap, and the identical sister chromatids are forcibly dragged to opposite poles of the cell. Finally, in telophase and cytokinesis, the nuclear membranes reform, the DNA uncoils, and the cytoplasm splits in 2. By separating identical chromatids, mitosis serves as the default cloning mechanism. It strictly preserves the 2N genetic baseline required to build and repair somatic tissue. Gametogenesis requires a different approach entirely. Meiosis occurs exclusively in germ cells and introduces a massive structural divergence. A single round of DNA replication is followed by 2 separate rounds of cellular division. The distinction is immediate. During Prophase I, homologous chromosomes, one inherited from each parent, physically intertwine to form a tetrad in a process called synapsis. While intertwined, the chromatids break and exchange equivalent pieces of DNA. This crossing over event physically unlinks genes on the same chromosome, providing the mechanical engine that drives Mendel's second law of independent assortment. This structural pairing changes the geometry of the next step. In mitosis, individual chromosomes line up single file, but in metaphase I of meiosis, the homologous pairs line up side by side across the metaphase plate. As a result, anaphase I separates the homologous chromosomes, not the sister chromatids. This is a reductional division, cutting the ploidy in half to create haplite cells. Meiosis II follows immediately and operates as an equational division. It behaves exactly like mitosis, pulling apart the remaining sister chromatids without any further change to the ploidy. By intentionally breaking the rules of exact replication and reduction, Meiosis generates 4 distinct haploid gametes. This calculated variation introduces the immense genetic diversity required for a species to adapt and survive. Looking at the male reproductive system in this diagram, we can trace the continuous pathway of spermatogenesis. Sperm develop inside the highly coiled seminiferous tubules of the testes before passing to the epididymis to gain flagellar motility. The seminiferous tubules rely on 2 specialized cell types. Sertoli cells physically nourish the developing sperm, while the adjacent interstitial cells of Leydig secrete testosterone. Male gametogenesis is designed for volume and continuity. A single diploid spermatogonium yields 4 equal, fully functional sperm, producing millions of gametes every day from puberty onward. This diagram illustrates the female reproductive system, where oogenesis takes a highly staggered, finite approach. All the oogonia a female will ever possess are formed during fetal development. The meiotic divisions here are characterized by unequal cytokinesis. The cell distributes nearly all its cytoplasm and organelles to a single massive ovum, leaving behind resource-starved polar bodies that cannot function as gametes. The timeline is governed by strict physiological arrests. By birth, all primary oocytes are frozen in Prophase I. It takes until menarche for 1 oocyte per month to resume the process and complete the first meiotic division. Even then, the resulting secondary oocyte immediately arrests in Metaphase II. It will only complete the final stage of meiosis if it is successfully penetrated by a sperm cell. While the male reproductive strategy relies on a continuous, high-volume output, the female system is heavily gated. It uses specialized physiological arrests to concentrate resources and ensure maximum cellular quality for a single viable zygote. This graph maps the cycle's chemical software, driven by GnRH triggering pituitary FSH and LH. During the follicular phase, rising FSH and LH stimulate follicles to produce estrogen, rebuilding the endometrium. Approaching day 14, high estrogen switches to positive feedback. This switch causes a massive spike in hormones. The resulting luteinizing hormone surge physically triggers ovulation, releasing the ovum into the peritoneal cavity. After ovulation, the cycle enters the luteal phase. The ruptured follicle transforms into the corpus luteum and begins secreting progesterone. While estrogen rebuilds the uterine lining, progesterone is required to maintain it for potential implantation. If fertilization does not occur, the corpus luteum loses its LH stimulation and atrophies. Progesterone levels drop, the uterine lining is sloughed off, and the block on GnRH is removed to start the cycle over. Human reproduction operates as a highly specific mechanical sequence. It relies completely on these interlocking chemical thresholds and feedback loops to function. We can now see the entire architecture. Genetic accounting dictates the cellular ploidy. Specialized anatomy provides the physical hardware, and a 28-day hormonal cycle acts as the timing software. The MCAT tests your ability to disrupt these systems. If homologous chromosomes fail to separate during anaphase I, you must be able to predict exactly how that non-disjunction alters the ploidy of the resulting gametes. Consider the systemic impact of oral contraceptives. By artificially supplying estrogen and progesterone, these pills leverage the body's own negative feedback loops to suppress GnRH, completely blocking the mid-cycle LH surge and preventing ovulation. Success on this exam requires viewing biology as an interconnected machine. When you understand the specific function of every deer, you can accurately predict the exact mechanism that causes the entire system to fail.